hopefully more people will show up. So thank you everyone for being here today. This is the first AWMA webinar Lunch and Learn of 2026. Sorry we were delayed. Lots of stuff's happening. But today we have a great speaker. This is Dr. Danica Lombardozzi. She's an assistant professor in the Department of Ecosystem Science and Sustainability at Colorado State University. And she's also a project scientist in the Climate and Global Dynamics Lab at the National Center for Atmospheric Research. Dr. Lombardozzi leads the development of the agriculture module in the community land model and co-founded the Ozone Pollution Education Network, where she directs community science efforts at Ozone bioindicator gardens across the network. She earned her BA in environmental science from Colorado College and a PhD in ecology and evolutionary biology from Cornell University. So if you guys have any questions throughout the talk, feel free to type in the chat or raise your hand. But I think let's try and leave raised hand questions till the end. We'll have plenty of time for Q&A. And what else? I think those are it. Make sure to keep your microphone muted unless you're speaking. And I will let Danica take it away.
Yeah, thank you. And just as I mentioned before, I can't actually see the meeting right now with my screen in presenter mode. So if there are questions that come up that you think I should stop and answer, Annarelli, please feel free to jump in and interrupt me.
I will.
Yeah, so I thank you for the lovely introduction, Annarelli. And today I'm excited to talk to you about something that people don't think about very often, which is how ozone can affect plants. And so I've titled this talk, What Plants Reveal About Ozone, Visible Clues to an Invisible Air Quality Problem, because those plants can provide us visible clues, among other things. So I'll talk about this aspect of my research. And I guess I just want to highlight that I'm broadly interested in the intersection of human activities, plant processes, and Earth's atmosphere. And within the Earth system, all these pieces are connected and they all affect one another. And as you likely are aware, carbon dioxide concentrations are increasing. And the reason that I'm showing you this is the Keeling curve, which is CO2 concentrations at Mauna Loa in Hawaii. And the reason that I'm showing you this is because not only are humans increasing the concentrations, which is evident from this graph, but all of the oscillations that are part of that annual cycle are driven by plants. And so in the growing season each year, plants leaf out like they're doing right now here in Colorado's Front Range, and they start to photosynthesize. So they start to take carbon dioxide from the atmosphere, and that carbon dioxide that they take from the atmosphere causes this drawdown in carbon dioxide that you can see in the annual cycle of CO2 every single year. And so you can see that carbon dioxide concentrations peak in about April or May, just before the leaf greening really starts up in the Northern Hemisphere, and then really they start to draw down through September, and then starting in October and November, CO2 concentrations start to increase again as the Northern Hemisphere plants senesce. And you might ask, why the Northern Hemisphere? And it's just because there's so much more land mass in the Northern Hemisphere, and there's a lot of land mass in high latitudes, both temperate and boreal plants that are really driving this strong seasonality in CO2 cycle. And so the reason that I'm highlighting this is because plants have a large and measurable impact on Earth's climate. So my research really focuses on how terrestrial ecosystems respond to and contribute to environmental change. And I use a lot of different techniques. I use global scale models, I use plant observations, and try to integrate all of these processes to understand answers to these questions. So I want to I want to get started with thinking about what is ozone. Ozone is a molecule of 3 oxygen atoms, and it's found throughout our atmosphere. Most people commonly think of ozone as being in the ozone layer, where it protects us from sun's harmful UV radiation. And this is a benefit to humans. In fact, life as we know it would not have evolved on Earth without this ozone layer. But the same exact chemical compound, ozone, found at the ground level, where in the air that we breathe, can be toxic to humans and to plants. And so the term ozone can be confusing because of these different functions in different parts of the atmosphere. But where we breathe, or where we exist, humans, plants, animals, that ozone can be toxic and harmful to us. So ozone is a primary component of smog and is one of EPA's criteria air pollutants. And it's a tricky pollution problem because there's no such thing as a direct emission. Instead, ozone is formed in the atmosphere. This is a very simplified equation, but it's formed through the reaction of nitrogen oxides, volatile organic compounds, or VOCs, and sunlight. And sunlight is really important because that catalyzes the reaction that forms ground level ozone. Because ozone is harmful to humans, EPA has limits the amount of ozone that's in the atmosphere. And so the EPA limit is 70 parts per billion over the course of 8 hours. And this figure here, with all of these dots, show different ozone monitors where any of the dots that are in yellow, orange, red, or pink are areas where that are out of compliance with EPA's standard, ozone standard. And I just want to zoom in here on the region that we are in, the Southwest US. And you can see that we're right about the national, at the national average, or the national standard at 70 parts per billion. But we are seeing an increase in ozone concentrations here in the Southwest US. Between 2010 and 2024, there's been a 6% increase in average ozone across all the sites in the US Southwest. And if you look at Colorado's Front Range, you notice that since 2000, most sites have exceeded EPA's standard. So the EPA standard here is shown in the dotted line. And so for all of these different sites, EPA ozone monitors, you can see that almost all of them are out of compliance. And actually, since approximately 2017 or 2018, ozone concentrations have been increasing in this region at most sites. So this is a problem because, as I mentioned earlier, ground level ozone impacts our health. So the picture on the left shows a lung that's exposed to air with no ozone on the top, and then on the bottom there's a lung exposed to ozone, air with ozone, and what you can see is that the lung in air that was exposed to ozone is it almost looks sunburned. It's bright red, it's inflamed, and so it just gives you an indication of what ozone exposure can do to lungs, and it increases the incidence of asthma and other diseases, other respiratory diseases. So that's one reason why we care, and one reason why EPA regulates it as the 6 criteria air pollutants. But what is less commonly thought about is the fact that ground level ozone also impacts plants. And so this is an image of a snap bean plant that is particularly sensitive to ozone. We would call this plant a biological indicator, which means that it biologically indicates something about the air. And so this is an ozone bioindicator. And you can see that all the brown spots and even the portions of the leaf that are turning yellow underneath, that's all due to ground level ozone damage. And this is just exposed to ambient air here in Boulder, Colorado, at one of my ozone gardens. So that's a little bit of background on ozone and why we care. What I want to talk to you about today is, how does ozone damage to vegetation impact leaf scale physiology, global carbon and water fluxes, and climate and air quality? And then I'll talk to you for a little while also about ozone bioindicator gardens and the national network of gardens that we have. So let's start with how ozone damages leaf physiology. This is a cross-section of a leaf, and what is illustrated here is that carbon dioxide enters a leaf through the stomatal cells, and those stomatal cells are really specialized cells that regulate the size of the opening of the pore, the size of the pore or the opening on a plant leaf. And this responds to a variety of environmental cues like light or humidity or even the amount of carbon dioxide that's in the air. And so that carbon dioxide is getting inside the leaf and it travels to the site of where the chlorophyll are, the chloroplasts, and it's biochemically turned, that carbon dioxide is turned through biochemical processes into sugars through the process of photosynthesis. And so you can see that photosynthesis is regulated by stomatoconductance and this biochemical carbon fixation. And at the same time, there's water inside all that is in all the intercellular spaces within a leaf, similar to water that is in intercellular spaces in human bodies. But that water, when those stomatal cells are open, that water is evaporating out. And I guess I should say that the size of the pore is what we call conductance. That's the stomatal conductance. And so as carbon dioxide is entering the leaf to get fixed into sugars through the process of photosynthesis, that water is leaving. So think about carbon in, water out. And that's really what the stomatal cells are trying to regulate, is they're trying to maximize carbon gain for water loss. But I wanted to know how ozone affects these processes. And the reason is because anything that is in the air can actually get into the leaf surface. And so ozone enters the leaf the same way that, that carbon dioxide enters the leaf. So I wanted to know how it affected these processes. And so I set up a chamber experiment. This is an open top chamber experiment in an old field in upstate New York. And I used this combination of manipulative open-top chamber experiments and also data synthesis techniques to determine the responses of photosynthesis and stomatal conductance to ozone across a variety of species. And what I found is that both photosynthesis and stomatal conductance decrease. And so in this graph, the x-axis goes from 0 to 100, and this is percent of control. So anything that is at 100% means that there's no difference between the ozone treated plant and the control plant. And anything that is less than control or less than 100%, that's the reduction. What you can see is that the mean photosynthetic change is 21%. In most plant species, photosynthesis is reduced more than stomatal conductance. The mean change in stomatal conductance across these species is 11%. You'll also note that there are numerous types of plants that are represented in this analysis, and I grouped them into crops, temperate deciduous trees, and temperate evergreen trees. One thing that is worth noting is that 95% of the available data are from temperate ecosystems. So we don't know what happens outside of temperate ecosystems. But this information is still pretty informative. And really what is happening is that, and the reason between the disconnect or the differences in response of photosynthesis and stomatal conductance is because ozone, when it enters the leaf, it damages the processes that are regulating that biochemical carbon fixation. So that initial damage is to biochemistry, which has immediate effects on photosynthesis, but not necessarily on stomatal conductance that's regulating that water loss or the process of transpiration. Ozone does affect stomatal conductance and therefore transpiration, but it's just not as large of an impact as photosynthesis due to that direct damage to biochemistry. The reason that this is problematic is because when we're calculating stomatal conductance in models, our theoretical understanding relies on the fact that photosynthesis is one of the main terms that's regulating stomatal conductance. And so you can see from this equation that if we calculate stomatal conductance based on that damaged photosynthesis rate, we're going to overestimate the damage to stomatal conductance. And just on the bottom here, you can see how stomata are damaged by ozone. These are electron spectroscopy images of stomata due to ozone damage. And so you can see the healthy stomata on the left, and then all the way on the right is severe damage to the stomatal cells. So again, the fact that we use photosynthesis to calculate stomatal conductance means that we likely overestimate stomatal conductance responses. And so that's what I'm showing here is these calculations of both photosynthesis and stomatal conductance. And what you'll note is the model with no damage is just that gray line. And so when we are simulating photosynthesis and stomatal conductance and not accounting for ozone damage, we're overestimating the amount of carbon gain and water loss between leaves and our atmosphere. The black line shows the observations from my open top chamber experiment. What that red line shows is the photosynthesis. So when photosynthesis is reduced due to ozone damage based on the results of the study that I showed you previously. So you can see photosynthesis on the left and stomatal conductance on the right. When we apply this method where we reduce photosynthesis, we capture the photosynthetic observational responses to light, which is along the y-axis in that left panel. We capture the observational responses pretty well, but in the right panel, where we're looking at stomatal conductance, when we reduce photosynthesis, we're reducing stomatal conductance too much compared to observations. And so this is problematic because we're not capturing the water loss correctly. And so when we modify these equations so that we are adjusting stomatal conductance separately from photosynthesis, we get this blue line for stomatal conductance that you can see in the right-hand panel here. And that independent method where we're modifying stomatal conductance independently of photosynthesis shows a much better match with observations, although we're still overestimating the decrease in stomatal conductance a little bit, but this is much better than the overestimation of stomatal conductance responses due to photosynthesis.
Yeah, back to back Jan meeting.
Everybody have their microphone on. Thank you.
Yeah, so just to recap, ozone damage changes leaf physiology by reducing photosynthesis and stomatal conductance, but there are different rates of change that alter leaf carbon and water fluxes. So how does ozone damage then change global carbon and water fluxes? So I wanted to understand the answer to this question. So I used the algorithm that I developed for that leaf scale model to separate the impact of ozone on photosynthesis and stomatal conductance in a global scale model. And for this work, I used the community Earth system model. And specifically, I focus on just the land portion of this model. And so this is a large scale Earth system model that is widely used to understand climate change and Earth system changes. And we can run that land component model as a standalone model. It can also be coupled back to the atmosphere or the full Earth system. And so I just am highlighting here that I'm only using the land model and we're reading in data, a data-derived observational atmosphere to run this land model. And so I use the algorithms that I developed for adjusting photosynthesis and stomatal conductance to ground level ozone. And the ozone damage depends on the ozone concentration, the length of exposure, and how much ozone is getting inside the leaf through that stomatal conductance. So what I found is that there is a decrease in photosynthesis due to higher ozone concentrations. So this is a direct decrease in carbon gain. And the significance is that ozone reduces the carbon stored in our ecosystems, and that regions with the largest reductions are really in agricultural hotspots. And so if you look at the eastern and central United States, and also parts of Europe and parts of East and Southeast Asia, these are agricultural hotspots, and that's where we're seeing some of the largest decreases that are up to between 15 and 25% reductions in carbon gain. And so this has direct implications for agricultural yields as well. I forgot that I highlighted those spots. At the same time, we see a decrease in transpiration due to ozone. And so this is a direct decrease in water loss back to the atmosphere. And the significance here is that it can reduce evaporative cooling and increase our surface runoff. And also, I just want to highlight that it can decouple that carbon and water cycling. And so it reduces the efficiency, what we call the water use efficiency, or the efficiency of the plants gaining carbon for the water loss. And this, you can see this really clearly in the eastern and midwestern US, where decreases in carbon gain are up to 25% in some regions, but the decreases in water loss are only 15%. So this has ramifications for hydrology, and that's because plants get water from the soil. The water travels from the roots through the stem and into the leaves, and then it evaporates from the stomata, and this is the process of transpiration. So if ozone reduces transpiration, what happens to the amount of water in the soil? So imagine that those arrows coming out of the leaves are reduced. It's going to reduce that whole flow of water and likely increase the amount of water that's in the soil. And that is in fact what we see. So this is change in runoff due to ozone. So runoff is the flow of water over land that is not absorbed by the land. And I think one of the main things that stands out to me here is this increase in runoff due to the decrease in transpiration. And you can see this really well in the Mississippi River Basin, where we get increases in runoff of about 10% due to that change in transpiration. So ozone decreases carbon gain and water loss, especially in crop regions, and this can result in changes in hydrology, including increased surface runoff. So the whole Earth system is connected, and that means that leaf ozone damage could also potentially change climate and air quality. And so for this particular set of analyses that I'm going to show you, all of the model simulations were using that land model, that global land model connected to the atmosphere model. And I will just highlight that without the use of models, it's really hard to know and identify the actual, the direct impact of ozone on vegetation that affects the atmosphere and how that affects the atmosphere. You really can't disentangle those atmospheric and climate feedbacks without the use of a model. There's not really observational methods that can do that. And so what we found is that when you reduce stomatal, when ozone reduces stomatal conductance, that can increase surface temperature. And that's because you have lower stomatal conductance, so you have less evaporative cooling. And when you have less evaporative cooling, that results in warmer temperatures. And so these temperatures presented here are in Kelvin, which is the same thing, or approximately equivalent to Celsius, but this is up to 3 degrees Fahrenheit in warming due to plant ozone affecting plant processes. So here, just so you know, we're only looking at the effect of transpiration. We're not actually accounting for changes in leaf area due to ozone or any of the biogeochemistry. And we're looking at the response of land surface temperature just due to that changing stomatal physiology. So at the same time, we also found that plant ozone damage can increase ozone concentrations by up to 6 parts per billion in the Northern Hemisphere. And these effects tend to be largest in Europe, but they are also large in North America and other parts of the world. And this is due to 2 effects. The direct effect is the reduced stomatal uptake. So you have less ozone entering the leaf through the stomata. So that's one. So that means more ozone is staying in the atmosphere. So that increases atmospheric ozone concentrations. And then there's also an indirect effect where you have less evaporative cooling, so you have warmer temperatures, and that can lead to more biogenic isoprene emissions, so isoprene emissions from plant processes. And that leads to atmospheric chemical reactions that lead to more ozone in the atmosphere. So I know that this is a pretty quick tour of this research, but to summarize, ozone damage to plants reduces leaf photosynthesis and stomatoconductance. That can cause changes in global carbon and water fluxes. And then that leaf damage, because of the changes in carbon and water fluxes, can increase air temperatures and degrade air quality. And then those changes, the increased ozone concentrations, for example, can then feed back to further damaging plants through higher ozone concentrations. So this work really just highlights how observations of leaf level processes can be used to inform models, as well as how models can be used to scale processes and understand atmospheric feedbacks. It also really emphasizes the really important role of ozone damage on vegetation for a lot of other things that impact human health as well as vegetation. So next I want to dive into ozone bioindicator gardens, and this is a really fun project where we are using plants as indicators of local air pollutions. So I linked to the ozone garden website at the bottom here, or at the bottom of the slide. And this picture is a leaf of a cutleaf coneflower plant. And so these are plants that grow throughout the United States. This particular plant originated from seeds from cutleaf coneflower in Rocky Mountain National Park, and the National Park Service gave us permission to harvest some of these seeds and grow them in our own ozone bioindicator gardens. And so this plant was grown at the National Center for Atmospheric Research, just in front of the building in ambient air. And you can see that all the dots on the top surface of the leaf, those black, brownish black looking dots, that's all ozone damage on the leaf. So the gardens, all of the ozone gardens are in public places. This is the ozone garden at the National Center for Atmospheric Research, or NCAR, in Boulder, Colorado. I planted this garden first in 2014, and it is still going strong. You can see the signs that we have there and the garden in the front. And our garden network, in addition to planting the garden, I co-founded the garden network, which is currently over 40 gardens throughout the United States. And we actually have a few gardens internationally that are, there's some in the UK and there's some in Norway. And I'm hoping that we can figure out ways to expand this network internationally, but the focus right now is throughout the US. And you can see that there's a high density of gardens in Colorado's Front Range, just in part because that is where I can help support gardens the best, but they're all over the US, looking to add more locations all the time. It's not hard to get involved, and I can talk about getting involved at a later point in this talk. I also just want to highlight, though, that if you look back at this map of high ozone concentrations, and keep in mind that the yellow, orange, red, and pink dots are places where the ozone concentrations are exceeding EPA standards, and we do have gardens in many of those places. So Colorado's Front Range would probably benefit us to have a few more gardens in California. There's some in Salt Lake City, Utah. Again, maybe having some planted in Arizona and in Texas, and in that sort of Texas, Oklahoma, Kansas area would be really beneficial, but we also have them along the eastern seaboard and along the Great Lakes region. We have a variety of different gardens. These are 4 of the gardens that have been planted in Boulder, and I use these photos to illustrate how different gardens can look. And so some gardens are planted in containers, and other gardens are planted directly in the ground. There are all sorts of gardens. There are many different ways to be able to plant these plants. And so it's an exciting opportunity because you can have a garden that is a small container garden, or you can have a garden that is much more expansive. It can be planted directly in the ground, or in the case in the NCAR front entrance, it's planted in a very, very large planter box. The one thing that I would say with gardens planted in pots, though, is that you have to make sure that they're well watered, because drought stress can cause plant stomata to close, and that can reduce the amount of ozone that plants are seeing because less ozone is getting inside the leaf. So drought stress can protect from ozone stress, but it doesn't mean that the plants are not hurting in that way. The ozone stress is just secondary to that drought stress. Here's a few more images of the gardens and all of the people that tend to visit the gardens. We had some high school interns that came to help with some of the gardens. We have public tours and programs that visit the gardens at NCAR. There's some elementary school kids. So the gardens can reach a range of people across different ages. One of the things that I think is the most exciting is just watching how people get excited to look for this damage, and also trying to improve our understanding of the difference in ground level ozone and stratospheric ozone. That is one of the most common misconceptions that I hear people have when we talk about ozone. They think that ozone, and it is, but they associate ozone with the beneficial ozone layer, and that any damage due to the damage in the ozone layer, which is not quite correct. And so this is really a tool to, in addition to collecting data, which I will talk about momentarily, but it's also a tool to help people improve to help improve public understanding of ozone pollution and the pollution problem. So they can look at these plants and they can visibly see that impact. So these plants kind of act as a canary in the coal mine, where they are indicating that our air is unhealthy by showing these visible signs of damage. And so these are the 4 plants that we plant most commonly in our ozone gardens, and you can see the ozone damage on each of them. So on the left is common milkweed, and you can see that all the black spots, those are it's all ozone damage. On the coneflower, which is second from the left, you can also see a lot of spots. Those are more discrete spots as opposed to spots that are congealing, like on the milkweed. The potato also gets kind of larger spots on its leaf. The snap bean, again, also gets reasonably large patches of brown. And so the ozone damage looks a little bit different on each of these different plants, which is why it's important to know what it looks like on the plants. But it all starts off the same way. It starts off as small pinpoint spots on the top surface of the leaf, so similar to what you see on the coneflower, and those are called stipples. Ozone damage typically occurs on the top surface of the leaf. You're not seeing it all the way through to the bottom of the leaf until the damage gets extreme enough that you're seeing dead patches on the leaf. So it starts off as that dark stippling on the top surface of the leaf. And another really characteristic feature of ozone damage is that it doesn't cross the leaf veins. And so if you look at the milkweed picture and where that damage is, you can see those bright veins still sticking out in the patches of black or the areas that have been severely impacted by ozone. So just to more clearly illustrate or I guess walk you through identifying ozone injury, it starts as stipple. It typically occurs on the top surface of the leaf. It only occurs between the leaf veins. It accumulates through time. That's another thing that I did not say. And because it accumulates through time, it affects older leaves more. And so the pictures that you can see here is a progression of ozone damage on the potato plants. July 2nd, we had of this particular year, we had very healthy looking potato plants with no ozone damage. On July 24th, I started to see the first signs of ozone damage. And so you can see in the sort of right part of the picture, there's one small dot. That's the first sign of stippling on this potato plant. And so it takes a pretty close look to be able to see that. And then at the bottom, you can see September 3rd, the ozone damage is much more severe and much more extreme. We also have resources on our website to help you learn how to identify ozone injury. And so this walks you through each stage of ozone injury. And then we also have a training game where you can start to see if you can pick out the damage. And so there's also a photo gallery that you can look at that shows what ozone damage looks like and what ozone damage does not look like. The reason that we know what ozone damage looks like is because of experiments like what I did for part of my dissertation, which was to run these open top chamber experiments. And so you can see this experiment where I was blowing high ozone concentrations into half of these chambers, and the other half was seeing just ambient air. And then here in the lower right is a picture of the tulip poplar. It's one leaf of the tulip poplar plant that was in one of these ozone exposure chambers, and you can see just how much this leaf is damaged from ozone due to these higher concentrations of ozone. Again, you can see the stipples, so the dark spots on the top surface of the leaf, and the bright green veins sticking out from the leaves. There are things that mimic ozone damage. That's why it's important to understand what ozone damage looks like. The things that mimic ozone damage are things like on the left here, it looks a little bit like stipples because they're dark, independent, like discrete dark spots that are round. But if you look closely, you can see that some of those dark spots are occurring kind of on the leaf vein, on that central vein. And this is what gave me pause. Also, these spots look too large for what ozone stippling usually is on milkweed. And when I rubbed the leaf, I realized that it was actually these spots are actually insect poop that rub off. Stipples don't rub off. On the left is a potato leaf, and this is the white patches are damage from a leafhopper insect that kind of chews out the top surface of the leaf. And you can see that these are lighter in color and they're irregularly shaped damaged patches on the leaf. And so this is also not ozone damage. We invite everyone in our gardens to collect data. People can collect data. We ask them to collect data on 10 leaves of an individual species if they're going to collect data, but you can collect data on more leaves and on more than one species, depending on which ozone garden you're visiting. And we categorize the ozone damage into 6 different categories. The first category being no ozone damage, the second category being 1 to 6% ozone damage, the third category between 7 and 25%, and so on up to Category 6, which is 75 to 100% of the leaf is damaged by ozone. And in collecting data, you're helping us to better understand when ozone damage is occurring, where it's occurring, and how severe it is. And so one example, these are some high school interns that I worked with at NCAR who did an analysis of ozone data at the Purchase Knob Garden in North Carolina. This is the longest running ozone garden in the US. And so what this is showing is the proportion of the leaves injured and the injury scores. So you can see that the colors correspond with the categories. And I want to highlight 2 things here. One, if you look at this 2010, it's a little clearer, but you can see that the damage gets more severe as you go further along into the growing season. And so you can see that in July, there was not much ozone damage. Only a small percentage of the leaves had any ozone damage at all. And then by September, that ozone damage was much more severe, with some leaves reaching that highest level of damage category, and more than half of the leaves having at least 25% damage. Another thing that stands out is that we have low ozone years and high ozone years. And in low ozone years, we don't see as much damage as we do in high ozone years. And so this is another thing that data collection at these gardens is helping us to understand, is that year-to-year variability. I am also particularly interested in the different responses of the different plants, because I think what I have noticed anecdotally, but I don't yet have data analysis to back this up. But what I have noticed is that the crop plants that we have, so the potato and the snap bean, they tend to have ozone damage that happens at more of a threshold. So they have no ozone damage, and then all of a sudden you see the first stipple, and within a week there's pretty severe ozone damage on a lot of the leaves. Whereas plants more like the cutleaf coneflower, that ozone damage can start a little bit earlier and it just gradually increments. And the damage doesn't seem to have as much of that threshold response. So these are things that I'm trying to understand. Just this morning, I got some new figures from undergrad who's doing some research in my lab where she was showing me the ozone damage on different plant leaves. I just did not have time to update this presentation to incorporate those data, but I'm really excited to share those with you at some point soon. So the ozone gardens are made of, or they're planted with plants that can help to visually identify that air pollution. It can also help to raise awareness about ozone pollution, and another thing that we're trying to document and quantify is that the damage increases through time and can be linked to ozone concentrations. So this really is a work in progress. We're trying to better understand what ozone levels cause visible damage, how quickly does it develop, how do different plants respond differently, and where geographically throughout the US are we seeing the most damage? And is the most damage in places where ozone concentration is the highest? If you have two areas where ozone concentrations are similar, are there other environmental factors that are contributing to some of that damage? So those are all the types of things that we're trying to understand. So what we're doing to try and answer these questions is to collect data frequently at existing gardens so that we can understand that development of damage through time. We're trying to add more garden locations and ozone monitors so that we have a larger sample size and connect ozone damage with ozone concentrations. And then we hope to continue this project for many years so that we can better understand year-to-year variations in the same locations. We don't have very many gardens that have been collecting data for multiple years at this point, and so we're looking forward to adding adding data collection at these gardens over subsequent years. If you're interested in planting an ozone garden, we have information for you. I did not include it here, but we do have information for you. You do not have to collect ozone damage data in order to plant a garden, but I personally am very excited when gardens do collect damage because they help us answer these questions. But even without data collection, they're really a very useful tool to help the public better understand the impacts that air quality has, not just on humans, but also on plants. And being able to see an indicator of that ozone damage also helps people to better connect it to what might be happening to their health. So to summarize, ozone damage injures plant leaves. It can cause visible damage and it changes physiology, including photosynthesis and stomatoconductance. Those changes to physiology can change global carbon and water fluxes. And those changes in global carbon and water fluxes due to physiological changes can lead to warmer air temperatures and higher ozone concentrations. So I guess just to wrap up, ozone damage to plants changes physiology, it changes climate, and it changes air quality. And these impacts range from leaf to global scale. What can you do about ozone damage? There are a lot of things that you can do. These are 6 easy things. This was put together by the Regional Air Quality Council. But you can drive less and use public transit. A lot of these things you'll notice are going to be somewhat similar to things you can do to reduce your impact on climate. But other things include tuning up your car, making sure that tires are properly inflated, and again, using electric or fuel efficient cars. One thing that might not be as intuitive is refuel in the evening and don't top off. Why refuel in the evening? Well, ozone requires sunlight in order to form. And so if you refuel in the evening, you're not giving, we don't have as much time to for that ozone to form. Similarly, we can make sure that your cars are not idling for more than 30 seconds, do yard work in the evening and use electric equipment if possible. And that's similar to refueling in the evening in terms of emissions. You're just, the emissions have time to form something else besides ozone, just due to the fact that there's less sunlight in the evenings. and overnight. And then also choose low volatile organic compound paints, carpets, and other products. So with that, that's the end, and I can stop sharing and take any questions that you might have.
So, oh man, that was great. I'm so excited. I have a couple questions, but let's open it up for the audience. So, who has a question? Feel free to raise your hand or unmute. Kelsey.
Hi, this is Kelsey Simpkins. I'm with the Regional Air Quality Council. So, Danica, I just want to say thanks for the great plug at the end. I love this presentation, and I am doing the horrible, like this is a comment and not a question. Although I do have a very quick question. The fun fact is that just about 10 years ago, I started my graduate program at CU Boulder with the Center for Environmental Journalism. And one of our very first field trips was to come out to NCAR and hear you speak and learn about your gardens. And so I just want to make that fun connection. It's like 10 years later.
Yeah,
I'm running our Simple Steps Better Air program. And basically I am going to write a little blog about your work for our new website. So I'm going to follow up with an email, maybe for some fun images. But my very nerdy question, just briefly here, is would you consider, having also worked in the climate change arena globally, would you consider ozone to be causing a feedback loop basically at this point?
Yes. Yeah. And that was what I was trying to highlight on that that sort of summary of the plant level to global level responses is really ozone damage to plants can cause feedbacks to climate that then increases ozone concentrations that then leads to additional damage. And so it absolutely can cause those feedback loops. It's not, those feedback loops are not something that we can understand without using models. And so it's really hard to evaluate them, but we can evaluate a lot of the different steps along the way to better understand what the, how that damage works and it impacts each step along the way.
Yeah, and I'll just say thank you again so much. One thing I like to try to frame working on ozone in the Front Range with is that climate may seem like this really big, overwhelming global problem, right? Because it is, but working on ozone locally is like that first really important rung of a ladder that has both immediate health benefits to reduce as well as working on the bigger picture. So I loved hearing all of the information and the work you've done that connects all of that. Thank you.
Oh, great. Yeah, I'm glad to hear of your interest, and it's nice to reconnect with you.
I have a question in the chat by Dominique Rowell. Are there common plants along Colorado's Front Range outside of the gardens where you have noticed visible ozone damage?
Yes. So there's cutleaf coneflower all over Colorado, and I have seen ozone damage. In fact, I think I made somebody upset by visiting a community garden in Lyons and seeing their cutleaf coneflower, and I was so excited to go look for ozone damage, and they were not happy to hear about it. But I did find ozone damage on those plants, and part of the reason why we collected seeds from the plants in Rocky Mountain National Park is because there had been documented ozone injury on those particular plants. And so the seeds that we have for our gardens that were collected by Rocky Mountain National Park for us were seeds from plants that have that high ozone concentration or ozone damage, sorry. There's the National Park Service has done a lot of work in this space as well, and they put together a list of ozone bioindicator species. And I will have to look up that, I can look up a link to that list momentarily and see if I can find it quickly, but it's also a great resource. Other plants that are known bioindicators that I know grow in Colorado, but we have not actually found the damage in Colorado are ponderosa pine and quaking aspen. And I have been wondering if part of the reason why we're not seeing damage on the on populations here in Colorado. So we've seen ozone damage on quaking aspen, for example, in Wisconsin, and ponderosa pine in California. And part of the reason why I'm wondering if we don't see it here is because plants here are adapted to high light because we're higher elevation, and plants that are adapted to high light tend to have higher antioxidant concentrations, and antioxidants are one of the ways that plants defend themselves from ozone concentrations and ozone pollution. So that's my working theory right now. It's an untested hypothesis, but that's my hypothesis. I hope to test it one day if I can get funding to do so.
Cool. It's live science, people. Okay, that's awesome. Thank you. Okay, next question was from Weston Carlos.
Yeah, hi. Thanks for the presentation. It was really interesting.
Great.
I think from the photos, I definitely understood that some plants are more susceptible to ozone exposure than others. So my question is, are there any common structural features to the plants that are less susceptible? I'm thinking like some plants have leaves that are kind of waxy, so they lose less water. Is there anything like that or any other structural feature that is common among the plants that are more tolerant of ozone?
Yeah, well, I mean, perhaps structural, but that from a physiological perspective, lower stomatal conductances and higher water use efficiency. So when plants can better gain carbon and keep their stomata more closed, you're going to prevent ozone from getting inside the surface of the leaf. There hasn't been quite as much research done on deposition to the waxy surface of the leaf, but the few papers that I have seen that have looked at that show that ozone can damage that waxy cuticle, but I think it has to be at pretty high concentrations for that to actually damage the plant. The damage is mostly through entering the stomata of the leaves. And so the stomatal conductance is one major factor that confers resistance to ozone damage. And then another factor is plants' antioxidant capacity. And so ozone, one of the things that I didn't say but is important is that when ozone enters a leaf, it's the 3 oxygen molecules. And what happens is that one of those oxygen molecules breaks off. So it forms oxygen, like atmospheric oxygen, O2, and then one free oxygen radical. And that free oxygen radical is what causes the damage. And so the plants make antioxidants that can scavenge that free oxygen radical. And so plants that have higher antioxidant capacity and they can regenerate those antioxidants more quickly, those tend to be plants that are also less sensitive. I also didn't highlight that we have, so the variety of snap bean, for example, that we have has been maintained by USDA for our ozone gardens, and they will send us seeds for beans that are resistant to ozone damage and that are more sensitive to ozone damage. The resistant beans, they still do show signs of ozone damage, but not nearly as easily as that sensitive beans. And so you can actually compare, even within a species, there are like population and individual level differences that confer that ozone damage. Similarly, with the potato, most potatoes that are grown commercially these days do not show ozone damage because of that, because they have been bred to not be sensitive to ozone. And so this is a particular variety of potato called La Chipper potato that when it was used in field trials in the 1970s, it was so sensitive to ozone that it was actually just plowed back into the field because it was that was more cost efficient than trying to actually harvest the potatoes because the yields were so low due to that ozone damage. And so it's just not something it's not something that people continue to plant.
Awesome. And we have time for one more question from Denny Alapatu.
Hi, Professor. Thank you for that outstanding presentation and sharing the study with us. My question is related to the wildfires. Do you think that this impact of the ozone on the leaves will lead to early drying that way that can contribute to more severe wildfires?
That's an interesting question. I haven't ever thought about it in that way. The ozone will damage the leaf for sure, and what it does is it speeds up senescence at a whole plant level. And so it causes the leaves to fall off the plant more quickly. Sometimes it can almost act to it, I wouldn't say stimulate growth, but stimulate the growth of new leaves. But I would say overall the plants are not gaining as much carbon, so it's not really stimulating growth. But because the leaves are falling off so much more quickly, it's causing additional litter, for example, that's going into the soils, and maybe that could cause and change flammability. The leaves, so the stomatal responses that I showed here, which is they decrease ozone, so that can actually lead to slightly more soil moisture, and that was that runoff feedback that I illustrated. But it doesn't mean that the plants are healthier, and there have been some studies that have actually shown that the stomata become sluggish at high enough ozone concentrations, and so they stay open and they end up losing more water in the end. And so that could also potentially cause to drying that could lead to more susceptibility from wildfires. The way that I more often think about ozone in wildfires is the fact that wildfires produce ozone that then can damage plants. And that's like a downstream atmospheric chemistry impact of wildfires.
Awesome. Thank you so much. Thank you everyone for your questions. And this webinar will be posted on our website on the recording, so you can share it, or if you missed part of it, come watch again, and follow us on LinkedIn to know what the next webinar is. The next webinar is actually on indoor air quality on May 20th. And I think that's all I have for announcements for now. But yeah, again, thank you so much. Thank you, Danica. It was nice seeing you.
Thanks, Annarelli. It was nice to see you as well. Thanks for inviting me to give this talk.
Yeah, no problem. All right, everyone have a great day.
Take care, everyone.